Hi, this is Peter Voorhees from Atrium Health, Levine Cancer Institute, and the Wake Forest University School of Medicine. We did look at various T-cell markers and how they correlated with these long-term remitters, specifically those that made it to the 5-year mark and beyond without progression of disease. And what we found is that the ratio of T cells to neutrophils at the time of leukapheresis was associated with longer term better outcomes. We also saw that in the CAR T cell product itself, prior to infusion, that a higher number of CAR positive naive T cells was associated with these long-term remissions. And after infusion, at the time of peak expansion, a high proportion of T cells with a central memory phenotype was associated with longer-term disease control. We also saw that at peak expansion, just a higher number of CAR-positive T cells in general relative to tumor burden, or what we call effector-to-target ratio, was associated with better outcomes. And why that's important is that, one, we think that using something like Cilta-Cel earlier in the course of therapy, when the patients are less heavily pretreated, will lead to a higher proportion of patients with fit T cell products to use. We also think that using Cilta-Cel earlier in the treatment continuum affords us the ability to use therapeutics to control the disease before leukapheresis and after leukapheresis as the CAR T cells are being manufactured, thereby optimizing that effector-to-target ratio and potentially leading to even better outcomes in earlier relapse disease and in potentially newly diagnosed disease as well. Yeah, so I think, as far as this study is concerned, obviously we want to see what happens long term with these 33% of patients who remain disease-free at the 5-year mark, right? So what is that going to look like at 10 years, for example, at 15 years? And the continue study, which these patients are currently being followed on, is going to assess that particular question. So really, the tantalizing question is, are any of these patients functionally cured? And I think that remains to be seen. But again, no one would argue with the fact that these are the most impressive, results that we've seen in this patient population to date. I think what's really interesting to see is how this is going to play out in earlier relapse. Specifically, what are the long-term efficacy results in CARTITUDE-4, where patients had only had 1 to 3 prior lines of therapy, and even more importantly, in CARTITUDE-5 and CARTITUDE-6, which are studies looking at the use of Cilta-Cel in frontline multiple myeloma patients. How effective is it going to be there? And are there going to be an increasing number of patients who are, for all intents and purposes, cured? And again, getting to this idea of T-cell fitness and being able to better control this effector-to-target ratio, we think, utilization of something like Cilta-Cel for those patients could really have profound implications for long-term disease control.